In the quiet stillness of a late evening, let us begin a slow, unhurried journey into one of life's most fundamental mysteries. Imagine something so small that millions of it could fit onto the head of a single pin, something invisible to the naked eye, yet so intricately crafted, it contains the blueprints for all of life. That something is the cell biology's quiet architect. Every living organism you've ever known or heard of, from the blades of grass brushing against your ankles to the beating heart within your own chest, is made of cells. They are the basic units of life, the smallest structures capable of carrying out all the processes we associate with living things. To understand the living cell is to understand the foundation of life itself. Cells are not simply building blocks. They are active, dynamic entities. Within them, countless processes occur every second, all in silence. Nutrients are absorbed, energy is produced, waste is removed, and information is carefully copied and transmitted. And yet, you've never noticed it happening. That's because the world of the cell is profoundly quiet. It moves at a scale and speed that is nearly impossible to perceive without magnification and time. Still, its quietness does not mean simplicity. Quite the opposite. Let's ease our way into the world of biology by observing a single, living cell. Not under a microscope just yet, but through the imagination. Picture a gentle droplet of water suspended, clear, and calm. Floating within it, a small, soft-walled enclosure. This is the cell. Its boundary, the cell membrane, keeps the internal environment steady and safe. Inside, the liquid interior is called cytoplasm, a gentle, jelly-like substance that holds all the cell's components in place. These components, known as organelles, are each responsible for different tasks. Though minuscule, the cell is a complete system. Like a miniature city, it has a central library, power stations, delivery routes, and recycling centers. The library, the nucleus, houses the instructions that guide every activity. These instructions are written in the language of DNA, coiled delicately within. Around it, the other organelles perform their functions with quiet precision. The mitochondria create energy, the ribosomes construct proteins, the endoplasmic reticulum and Golgi apparatus sort and distribute resources. Each structure plays its part in maintaining the whole. And yet, cells are not uniform. Some are simple and self-sufficient, like the single-celled organisms that drift through ponds or soil. These tiny beings, such as bacteria, are prokaryotic, meaning they lack a nucleus. They are ancient, resilient, and elegantly pared down. They have survived for billions of years with a streamlined structure and continue to thrive in nearly every corner of the earth. Other cells are far more complex. These are eukaryotic cells found in plants, animals, fungi, and many types of algae. Their internal layout is more elaborate, with distinct compartments for different tasks. And yet, even with this added complexity, the principles remain the same. Absorb, process, respond, and replicate. It is in these cells that we find the foundation of our own bodies. Your skin, your muscles, your eyes all are made up of countless specialized cells, each carrying the same underlying instructions, yet adapted to perform a unique role. They form tissues, which in turn build organs, which combine to create the entire human form. But all of it begins with a single cell. From the moment of conception, when two cells merge to form one, a fertilized egg, a cascade of division, differentiation, and development is set into motion. From that first cell, the body unfolds, one silent instruction at a time. Cells do not act alone, however. They communicate constantly, sending chemical messages to one another, responding to changes in their surroundings, cooperating as part of something larger. When a cut appears on the skin, it is the surrounding cells that begin the repair. When you grow, it is because cells divide. When you feel heat or cold, cells transmit signals that your brain interprets. There is a kind of quiet wisdom in the way cells operate. They do not rush. They do not need to be told what to do. Their behavior is encoded in them not as thoughts or intentions, but as patterns refined over the course of evolution. The word cell itself was coined in the 1600s by Robert Hooke, who, upon examining thin slices of cork through a primitive microscope, noticed tiny compartments resembling the small rooms or cells occupied by monks. What he did not realize was that these cork cells were long dead, empty husks of what once lived. But the name endured. Today our understanding of cells has grown more precise and profound. We know that cells come in countless shapes and sizes, adapted to fit their roles. Nerve cells can stretch over a meter long. Red blood cells drift through vessels by the millions. Some cells possess flagella, long, whip-like tails that help them swim, while others remain anchored in place, forming part of stable tissues. Despite this diversity, all cells share three core features: a boundary that separates them from the outside world, internal material that carries out life's functions, and genetic instructions to guide them. No matter how different they appear, this shared architecture links all life together. And so, as we begin this gentle exploration of biology's smallest unit, we are also opening a door to understanding life itself, not through noise or spectacle, but through the deep calm that governs the inner workings of cells. In the parts ahead, we will delve further into the nucleus, the organelles, the processes of division and communication. But for now, let your mind rest on this single, quiet idea, that within you, and within every living being, there are countless tiny worlds, each one a cell, each one alive in its own right, performing the subtle symphony of life. Part 2. The origins of cells. As we drift further into the gentle rhythms of biology, our thoughts now turn backward, far, far back in time, long before humans, before animals, before even the simplest plants had taken root in the oceans. We return to a world where no eyes had yet opened, no hearts had yet beaten. A young earth, wrapped in volcanic clouds, warmed by a restless sun. The surface was unstable, with molten rock and boiling seas. The air was thick with gases we could not now breathe: methane, ammonia, and carbon dioxide. And yet, in this unlikely crucible, life began. The story of cells begins not with life as we know it, but with chemistry. In the ancient oceans, organic molecules began to form. These were the simplest building blocks: amino acids, sugars, lipids. They collected in puddles warmed by the sun, or drifted near underwater vents, where mineral-rich water seeped from beneath the seafloor. Here, through reactions not fully understood but endlessly studied, complexity began to build itself. Over time, some molecules began to arrange into structures. Fatty acids formed bubbles, small, round enclosures with watery centers and thin membranes. These primitive bubbles, called protocells, were not alive, but they hinted at something more. They could contain other molecules. They could separate their insides from the outside world. And in a world where chaos reigned, that simple boundary, a membrane, became a kind of protection. Inside some of these enclosures, other molecules gathered. Among them were RNA-like strands, chains, capable not only of storing information, but of copying themselves. This, perhaps more than anything else, marked the beginning of life. The ability to replicate, to create new copies of a structure, is what separates the living from the non-living. Once replication was possible, natural selection could begin. Tiny advantages, a more stable membrane, a more efficient enzyme, made some protocells more likely to survive and divide. And over time, those tiny changes added up. Protocells became true cells. The first living cells were simple. They were prokaryotic, lacking nuclei or internal compartments. Yet even in their simplicity, they were astonishing. These early cells carried out chemical reactions, maintained their boundaries, responded to their environment, and reproduced. In other words, they were alive. This all occurred some 3.5 to 4 billion years ago, long before multicellular life would appear. For a vast stretch of time, Earth was inhabited solely by these microscopic beings. Single-celled organisms ruled the planet in silence. Among them were bacteria and archaea, the two main branches of prokaryotic life. Though visually similar, bacteria and archaea are biochemically distinct. Archaea, for example, can survive in extreme environments, boiling springs, salty lakes, deep-sea vents. Their resilience suggests that early Earth may have been more suited to their hardy design. Bacteria, meanwhile, would go on to influence the course of life more profoundly than perhaps any other organism. Some learned to photosynthesize to capture sunlight and use it to create energy. In doing so, they began to release oxygen as a byproduct. This release of oxygen slowly transformed the Earth's atmosphere. What was once a toxic blanket of gases became breathable air. Over millions of years, this shift made possible the emergence of more complex life forms. But before that could happen, cells themselves would need to evolve. At some point, a remarkable transition occurred. Some cells began to develop internal compartments, wrapped in membranes. One of these compartments became the nucleus, the structure that now houses DNA in all plants, animals, and fungi. The origin of the nucleus and other organelles is explained in part by the endosymbiotic theory. According to this idea, some early cells engulfed other, smaller cells not to digest them, but to form partnerships. A bacterium capable of producing energy, once taken in by a larger cell, became the ancestor of mitochondria. A photosynthetic cell, once swallowed, became the origin of chloroplasts in plant cells. These symbiotic events created the eukaryotic cell, larger, more complex, and better able to support multicellular life. Eukaryotes, with their inner compartments and refined genetic control, had new possibilities. They could specialize, divide labor, and form colonies. Some of these colonies eventually gave rise to multicellular organisms where different cells performed different tasks, all working together as one. And so, the diversity of life began to blossom. All around us today, the legacy of this transformation continues. Every human cell contains mitochondria, remnants of ancient bacteria, still producing energy. Every plant cell carries chloroplasts, green echoes of ancient symbionts. Inside each nucleus, DNA coils in quiet spirals, preserving instructions that stretch back billions of years. It is humbling to realize that the cells in your own body share a deep kinship with the first cells that ever lived. They have changed, yes, but the core mechanisms remain. They divide, they respond, they pass along their genetic code. They are, in a quiet and literal sense, descendants of Earth's earliest life. Even now, countless cells drift through the oceans much as they did in the beginning, single, self-contained, and content to remain small. Their lives are simple, yet perfectly suited to the only task that matters in the long run: to survive and continue. And so, as we contemplate the origins of the cell, we see more than chemistry. We see persistence, quiet adaptation, the slow shaping of something that would one day give rise to trees, birds, rivers of thought, and the very voice inside your mind. The cell, though born in fire and water, now resides in the softest places in leaf and lung and fingertip. And as you rest, perhaps somewhere deep within you, millions of cells are quietly dividing, repairing, renewing, following a pattern written long ago, when life first stirred in the dark waters of an ancient world. Part 3. Prokaryotic Simplicity. Let us now focus our attention on the first true forms of life, the prokaryotic cell. It is a form so enduring, so efficient, that it has remained largely unchanged for billions of years, quietly persisting across every environment on Earth, from the frozen peaks of the Arctic to the searing interiors of deep-sea hydrothermal vents. Prokaryotes are the quiet elders of the biological world. A prokaryotic cell is simple in form, but not in function. Stripped of ornamentation, it lacks a nucleus. There are no ornate organelles here, no delicate membranes separating function from function, and yet, within its modest confines, it performs all the tasks that define life. The word prokaryote means before nucleus. These are the original cell types, the first biological structures to maintain themselves, reproduce, and adapt to their surroundings. They are typically small, most measuring between 1 and 10 micrometers. A single grain of sand could dwarf them, but this smallness is deceptive. Prokaryotes include two main groups, bacteria and archaea. Though they often appear similar under a microscope, they differ in profound biochemical ways. Bacteria are more familiar to us. They include species that live in soil, in water, and even within our own bodies. Archaea, in contrast, often inhabit extreme environments, salt flats, acidic springs, or near-boiling oceanic fissures. These archaea thrive where few others can, not because they are primitive, but because they are deeply specialized. In its most essential form, a prokaryotic cell consists of a cell membrane, a cytoplasm, and a loop of DNA. Most are also encased in a cell wall, a rigid structure that provides shape and protection. Unlike plant cell walls, which are made of cellulose, bacterial cell walls often contain a substance called peptidoglycan, a mesh of sugars and amino acids that creates a strong yet flexible shield. Within the cell, the DNA floats freely in a region called the nucleoid, not bound by a membrane, but gathered in gentle loops and coils. This DNA carries all the instructions for the cell's structure and behavior, a genetic code passed down through generations. Also within the cytoplasm are small, grain-like structures called ribosomes. These are the protein factories of the cell, translating genetic instructions into the building blocks of structure and function. Even without the specialized organelles of more complex cells, prokaryotes carry out the same fundamental processes. They generate energy, build proteins, replicate their DNA, and divide. Their method of division is simple, yet effective. Through a process called binary fission, a prokaryotic cell makes a full copy of its genetic material, grows in size, and then splits into two identical daughter cells. This process requires no complex spindle fibers, no mitotic stages. It is smooth, efficient, and remarkably fast. Some bacteria can divide every 20 minutes under ideal conditions, a rate of reproduction that can quickly lead to vast colonies. Beyond the basics, many prokaryotic cells possess additional features suited to their environments. Some are covered in tiny, hair-like projections called pili, which help them stick to surfaces or exchange genetic material. Others may have flagella long, whip-like tails that propel them through liquid environments with surprising agility. This mobility is not random. Many bacteria exhibit chemotaxis, a behavior in which they swim toward beneficial chemicals such as nutrients and away from harmful substances. It is a simple form of sensing, an awareness not of thought but of orientation. Prokaryotic cells may also form protective structures called endospores when faced with harsh conditions. These are dormant shells that can endure heat, radiation, and dryness for years, even centuries. When conditions improve, the endospore can awaken and life resumes. It is a strategy not of escape, but of endurance. Though often discussed in terms of disease, most bacteria are not harmful. In fact, many are essential to life as we know it. Some help fix nitrogen in soil, making it usable for plants. Others live in the human gut, aiding digestion and synthesizing vital nutrients. The human body is home to trillions of bacterial cells, collectively called the microbiome, which outnumber our own cells and quietly influence everything from immunity to mood. It is worth pausing here to consider that these tiny organisms were Earth's sole inhabitants for more than 2 billion years. During this long and quiet reign, they shaped the planet's surface, altered the atmosphere, and laid the groundwork for all future life. They were not waiting, they were thriving, and they continue to thrive in rocks miles beneath the Earth's surface, in the icy crusts of Antarctica, and inside the warm curves of your own lungs. Their simplicity is their strength. With fewer internal parts to manage, they are highly adaptable, able to mutate quickly, swap genes, and colonize new environments with ease. The exchange of genetic material between bacteria, known as horizontal gene transfer, is another quiet marvel. Unlike human reproduction, which relies on inheritance from parent to offspring, bacteria can share genes with neighbors. This has allowed them to develop resistance to antibiotics, digest unusual chemicals, and adapt to novel environments with breathtaking speed. Despite their ancient lineage and simple design, prokaryotes remain among the most dynamic life forms on the planet. So as you breathe and rest and let your thoughts drift, remember that these humble cells, unseen and often unconsidered, are everywhere. They were here before us, and they will likely remain long after. In their silent simplicity, they tell a story not just of survival, but of persistence, a story that began in the early seas and continues now, gently and endlessly, in the soil beneath your feet, in the water you drink, and deep within your very own body. The prokaryotic cell, though small, carries with it the echoes of life's first spark and the quiet rhythm of life, still unfolding. Part 4. The Rise of Eukaryotes. Let us now continue our calm descent into the architecture of life by turning to the eukaryotic cell, a more complex, spacious, and intricately partitioned world than the one we've just left behind. Where prokaryotic cells whisper simplicity, eukaryotic cells speak in soft orchestration. They are the builders of the visible world, the cells that form plants, animals, fungi, and algae. Eukaryotes emerged more than 1 billion years ago. Their arrival did not erase the prokaryotes, but added new dimensions to life's possibilities. These cells are larger, typically 10 to 100 times the size of a typical bacterium, but their defining trait is not their size, but their structure. The eukaryotic cell is compartmentalized, gently divided into smaller, membrane-bound spaces, each dedicated to particular functions. At the heart of this cell is the nucleus, a rounded structure wrapped in a double membrane, carefully protecting the strands of DNA held inside. This DNA carries the genetic instructions for the cell's function, growth, and reproduction. Whereas prokaryotic DNA floats freely, the eukaryotic genome is enclosed, shielded, and organized, bundled into linear chromosomes, coiled like threads around protein spools. This simple act of separation enclosing the genetic material transformed cellular life. With a nucleus came regulation, timing, and nuance. Processes could be staggered, separated, and controlled with subtlety. Instructions could be stored, copied, or silenced, all within the soft envelope of the nuclear membrane. Surrounding the nucleus is the cytoplasm, a semi-fluid matrix that cushions the many organelles' small, specialized structures, each wrapped in their own membranes, each with a task. Consider the mitochondrion, the powerhouse of the cell. These bean-shaped structures convert nutrients into usable energy through a process called cellular respiration. They take in oxygen and molecules like glucose and quietly release energy in the form of adenosine triphosphate, or ATP. It is this energy that powers all cellular activities, from motion to maintenance to repair. Mitochondria, curiously, carry their own DNA. They replicate independently, a quiet clue to their ancient past. Scientists believe mitochondria were once free-living prokaryotic organisms, taken in by a larger cell and gradually transformed from guests into permanent residents. This idea, known as the endosymbiotic theory, explains how eukaryotic complexity may have grown from cellular cooperation rather than conquest. A similar story applies to the chloroplast, found only in plant and algal cells. These green disc-shaped organelles perform photosynthesis, using sunlight to convert carbon dioxide and water into sugar and releasing oxygen as a gift to the surrounding world. Like mitochondria, chloroplasts carry their own DNA, another echo of their independent origins. Together, mitochondria and chloroplasts hint at a broader truth, that eukaryotic cells are not just more complex, but more collaborative. They are communities within a membrane, each organelle performing its role in service of the whole. Moving outward from the nucleus, we find the endoplasmic reticulum, or ER, a network of flattened sacs and branching tubules that weaves through the cytoplasm like an internal roadway. There are two types: rough ER, studded with ribosomes, where proteins are synthesized, and smooth ER, which helps build lipids and detoxify substances. These newly formed proteins and lipids are passed to the Golgi apparatus, a stack of membrane-bound cisternae. Here, molecules are modified, sorted, and packaged into small vesicles for transport, either to other parts of the cell or to the cell membrane for release. Elsewhere in the cell, small round lysosomes contain enzymes that break down worn-out cellular components, food particles, or invading microbes. They are the recyclers of the cell, ensuring that nothing goes to waste. In plant cells, vacuoles provide storage of water, ions, nutrients, or waste. A central vacuole can occupy most of a plant cell's volume, pressing outward against the cell wall and helping the plant maintain its structure. And beneath it all lies the cytoskeleton, a dynamic framework of protein fibers that gives the cell its shape, supports movement, and guides the transport of materials. It is not a rigid frame, but a living scaffold changing, flexing, adapting. Through it, organelles are moved along like passengers on invisible tracks. Unlike prokaryotic cells, which often live alone, many eukaryotic cells exist as part of a greater body. In multicellular organisms, cells do not simply perform all functions, they specialize. Some become neurons, long and sensitive, designed to carry electrical signals. Others become red blood cells, flattened and empty, ferrying oxygen through narrow vessels. Muscle cells grow long and fibrous, contracting in rhythmic pulses. Each form reflects its function. This specialization requires coordination. In the body, cells communicate constantly through chemical signals. Hormones, neurotransmitters, and local messengers allow distant parts of an organism to act in unison. A cut on the skin, a change in temperature, or the intake of food sets off a quiet cascade of cellular responses, each cell knowing its role, performing it without pause. Despite their complexity, eukaryotic cells retain the same core requirements as their simpler ancestors: energy, structure, replication, and regulation. The difference lies in their elegance, not in what they do, but in how smoothly and quietly they manage it all. So, as we rest with the idea of eukaryotic life, we begin to see the profound beauty in biological architecture, a design not rushed or crowded, but calm, layered, and remarkably organized. A membrane within a membrane, a message passed gently from one structure to another, each organelle speaking its language, yet part of a single, fluid conversation. The eukaryotic cell is not just a chamber of parts. It is a quiet symphony, each note purposeful, each rhythm steady, each pause filled with intention. And within your own body tonight, trillions of these cells go about their work, breathing, building, remembering, with no noise, with no rush, only the soft, steady hum of life carried out with grace. Part 5. The Cell Theory. Now that we have gently explored both prokaryotic simplicity and eukaryotic complexity, it is time to rest upon one of the most enduring foundations of biological thought, the cell theory. Not a single observation, nor a fleeting idea, but a quiet, guiding framework that continues to shape how we understand life itself. The cell theory, in its essence, is composed of three simple yet profound statements. 1. All living things are made of cells. 2. The cell is the basic unit of structure and function in living organisms. 3. All cells arise from pre-existing cells. These three statements, unassuming though they may seem, revolutionized biology. Before their acceptance, life was viewed through lenses of spontaneity and mysticism. People once believed that life could arise from non-life, that flies could be born from rotting meat, or that mold could simply appear from damp air. The idea that all living matter shares a common structure, that life begets life through ordered replication, was quietly transformative. To understand how this theory came to be, we must travel softly back to the 17th century. In 1665, Robert Hooke, using a simple compound microscope, examined thin slices of cork, the dead tissue of tree bark. Under magnification, he observed a pattern of small, empty compartments. They reminded him of the modest rooms inhabited by monks, and so he called them cells. Though Hooke could not see the living contents of these spaces, his naming gave birth to a concept. Later, Antony van Leeuwenhoek, a Dutch tradesman with a gift for lens-making, crafted microscopes of extraordinary clarity. Through these lenses, he observed tiny moving forms in pond water, what he called animalcules. These were bacteria and protozoa, living cells in motion, unseen by the human eye until that moment. His letters to the Royal Society described these discoveries with wonder, and the quiet world of the microscopic began to unfold. Still, it would take nearly 2 centuries before the pieces of the puzzle were placed together. In the 1830s, 2 German scientists, Matthias Schleiden and Theodor Schwann, observed plant and animal tissues under more advanced microscopes. Schleiden concluded that all plants were made of cells. Schwann extended the idea to animals. Together they proposed that all living things, regardless of form, are composed of cells, the first 2 tenets of cell theory. The third principle, that all cells come from other cells, was established by Rudolf Virchow in 1855. His phrase, omnis cellula e cellula, meaning every cell from a cell, quietly corrected the earlier notion of spontaneous generation. Cells, he observed, did not appear from nowhere. They divided. They passed on their contents. They continued the chain of life in unbroken succession. And so, by mid-19th century, cell theory had taken root not as speculation, but as careful observation woven into principle. These ideas may now feel obvious, so ingrained in textbooks and diagrams that we rarely pause to consider them. But within their quiet logic lies a universal truth. From the simplest bacterium to the vast networks of a whale's nervous system, all life is cellular. Every heartbeat, every breath, every thought you have ever had made possible by the activities of cells. The elegance of this theory also rests in its ability to unify. A redwood tree and a hummingbird seem utterly different, yet both are assemblies of cells. Each one begins as a single cell. Each one grows by the division of cells. Each one is sustained by cellular processes, the intake of nutrients, the creation of energy, the repair of damage, the expression of genetic instructions. The cell is the common denominator. It is the smallest entity that can be said to be alive. Molecules, no matter how complex, do not live. Viruses, though they reproduce and evolve, do so only by hijacking a host's cellular machinery. Only the cell meets all the criteria of life metabolism, growth, response to stimuli, and reproduction carried out in a self-contained, regulated environment. This is why the cell is called the basic unit of life. Within it, everything required for existence unfolds: a balance of ions across the membrane, a rhythm of energy production, a choreography of proteins folded and directed to their destinations, a constant scanning and repair of genetic material, none of it orchestrated by thought, yet all of it purposeful, quiet, unfailing. The cell theory also has profound implications for health and medicine. Disease, in this view, is not a curse or imbalance of humors, but a dysfunction at the cellular level. When cells grow uncontrollably, we see cancer. When they fail to respond to insulin, we see diabetes. When neurons degenerate, we witness memory fading or motion slowing. Each condition can be traced, gently, to something gone awry within the life of the cell. Likewise, healing depends on cellular renewal. Skin mends through cell division. Muscles strengthen through microscopic damage and cellular repair. Bones knit themselves together, one cell layer at a time. Even the medications we take, antibiotics, hormones, vaccines, work by interacting with cells or mimicking their signals. This centrality of the cell allows biology to be studied at multiple levels, all interwoven. A gene expresses a protein alters a function changes a tissue, and the tissue changes an organism, but the root remains cellular. And so, the cell theory offers both a beginning and a bridge. A beginning, because every living thing starts with a cell. A bridge, because through the study of cells, we connect molecular biology with physiology, anatomy with evolution, health with behavior. As we rest with this idea, we might picture the countless cells within us. Some are dividing, some are repairing, some are carrying oxygen, digesting sugars, or scanning DNA for errors. Each one has descended from a cell that came before, and each one will pass along its contents, as long as life continues. Not all theories endure, but this one has quietly, steadfastly, universally, and in its soft clarity, it reminds us that even the grandest expressions of life are, at their root, the work of countless tiny structures, performing their duties with silent grace, one cell, and then another. Part 6. Cell Structure Overview. As we settle deeper into the quiet intricacies of biology, let us now drift slowly through the architecture of a single eukaryotic cell, not hurriedly, but carefully, like walking through the soft, echoing halls of a living library. Each structure, though tiny, carries meaning. Each has a place, a role, a rhythm. We begin at the outermost layer, the cell membrane, also called the plasma membrane. This delicate boundary encases the entire cell. It is thin, flexible, and dynamic. Unlike a rigid wall, the membrane gently undulates and responds to its surroundings. It separates the inside of the cell from the outside world, but it is not closed off. Instead, it regulates, allowing certain substances in, guiding others out. This membrane is made of a phospholipid bilayer, 2 layers of lipid molecules with hydrophilic heads facing outward and hydrophobic tails tucked inside. Embedded within this bilayer are proteins. Some act as gateways, others as signals or anchors. The entire structure is selectively permeable, meaning it carefully chooses what to allow across. This is the first layer of life's regulation. Passing through the membrane, We enter the cytoplasm, a gel-like substance that fills the space inside the cell. It is not empty. It is alive with quiet movement. Organelles float gently in it, and countless molecules dissolve, diffuse, and react. The cytoplasm provides support, suspension, and a medium in which cellular processes unfold. Suspended within the cytoplasm is the most central of all organelles, the nucleus. The nucleus is often round or oval, enclosed in a double membrane known as the nuclear envelope. This envelope contains pores, delicate channels that allow certain molecules to pass in or out. Inside the nucleus resides the cell's most precious cargo, its DNA. This genetic material, organized into chromosomes, contains the instructions for making every protein the cell will ever need. When the cell is not dividing, DNA appears as chromatin, a loosely coiled structure that allows easy access for reading and transcription. Within the nucleus, there is also a darker region called the nucleolus, where ribosomes are assembled. Ribosomes, as we will see, are essential for building proteins. As we float away from the nucleus, our path leads to the endoplasmic reticulum, or ER, an extensive network of membranes winding throughout the cytoplasm. The ER comes in two forms, rough and smooth. The rough ER is so named because it is studded with ribosomes, giving it a grainy appearance under the microscope. These ribosomes are the site of protein synthesis. They read messenger RNA, mRNA, and assemble amino acids into long, folded chains. The rough ER, then, acts like a quiet factory building, folding, and packaging proteins, many of which are destined for export or use in the membrane. The smooth ER, by contrast, lacks ribosomes. Its roles are equally important but different. It synthesizes lipids, detoxifies chemicals, and stores calcium ions. In muscle cells, the smooth ER plays a crucial part in contraction by regulating calcium release. From the ER, newly made proteins are sent to the Golgi apparatus, a series of stacked, flattened membranes, often compared to a post office. Here, proteins are modified, sorted, and prepared for delivery. Some are tagged for transport to the membrane. Others are enclosed in small vesicles, lipid-wrapped parcels, and sent to various destinations within or beyond the cell. Close by, we find the mitochondria, the energy producers of the cell. These bean-shaped organelles have a double membrane and their own DNA. They take in nutrients, such as glucose, and use oxygen to produce adenosine triphosphate, ATP, the cell's primary energy currency. This process, called cellular respiration, occurs in stages, mostly within the mitochondria's folded inner membrane, known as the cristae. Mitochondria are highly active and essential. Without them, the cell would lack energy, and without energy, no biological process could proceed. In plant cells and some algae, we also find chloroplasts. These green organelles are the site of photosynthesis, where sunlight is converted into chemical energy. Like mitochondria, chloroplasts have their own DNA and internal membranes. Their green color comes from chlorophyll, a pigment that captures sunlight. They are the quiet gardeners of the cell, turning light into life-sustaining sugars. Scattered throughout the cytoplasm are ribosomes, either floating freely or attached to the ER. These tiny, dense structures consist of RNA and protein. Despite their small size, they perform one of the most important tasks in biology, assembling proteins. They read genetic instructions and translate them into function molecule by molecule. Another vital organelle is the lysosome, a small enzyme-filled sac that acts as the cell's recycling center. Lysosomes break down damaged components, invading microbes and worn-out molecules. They digest the unusable and release usable parts back into the cytoplasm. Without this cleanup system, the cell would quickly become cluttered and dysfunctional. Plant cells also contain vacuoles, large, fluid-filled sacs that store water, nutrients, and waste. In mature plant cells, A single central vacuole may dominate the interior, helping to maintain pressure and structure. It is a still reservoir, a gentle presence that keeps the cell firm and hydrated. And beneath all of this lies the cytoskeleton, a network of protein filaments that gives the cell shape and structure. It includes microfilaments, intermediate filaments, and microtubules, each with its own form and function. The cytoskeleton not only holds organelles in place, but also guides their movement. During cell division, it organizes chromosomes. During growth, it helps the cell stretch and reshape itself. In animal cells, two small structures called centrioles are found near the nucleus. These are involved in organizing microtubules during division, helping to pull duplicated chromosomes apart so that each new cell inherits a complete set. Together, these components form the living, breathing unit that is the cell. Each part has its role. Each works in harmony with the others. There is no central conductor. No voice shouts commands. Yet the cell operates with steady coordination, adjusting, responding, maintaining balance. This structure, though too small to see unaided, is the source of all growth, repair, thought, and motion. It is the scene of your development, your adaptation, your healing, and all of it occurs within this fluid, membranous chamber, where chemistry becomes consciousness, and where the blueprint of life is quietly brought to form. As we leave this inner landscape behind, let its elegance linger with you not as something abstract or foreign, but as something within you. Right now, this moment, your cells are at work folding proteins, sending messages, building membranes, and releasing energy, all in gentle silence. Part 7. The nucleus and DNA. At the quiet heart of the eukaryotic cell lies its most treasured space, the nucleus. Resting like a central chamber within the larger living structure, the nucleus is more than just a compartment. It is the cell's library, archive, and command center, all in one. Wrapped in a protective envelope of membranes, the nucleus holds the complete set of instructions that guide everything the cell does: when to grow, when to divide, what proteins to build, and how to respond to its surroundings. These instructions are written in a universal biological language, one shared by nearly every living organism on Earth. That language is DNA. Let us enter this space gently. The nuclear envelope consists of two concentric membranes, an inner and outer layer, that encircle the nucleus like a soft barrier. It is not sealed. Scattered across its surface are structures known as nuclear pores, which serve as guarded gateways. Through them, molecules pass in and out messenger RNA, leaving to deliver instructions, enzymes entering to assist with the work within. Inside, we find a fluid interior known as the nucleoplasm, where the chromatin floats a delicate tangle of DNA and proteins. When a cell is not dividing, the DNA exists in this loosely coiled state, accessible and ready to be read. Only during division does it tighten into the familiar X-shaped chromosomes. Each molecule of DNA is a double helix, a twisted ladder made of 4 chemical bases: adenine, thymine, cytosine, and guanine. These bases pair with one another in a predictable way: adenine with thymine, cytosine with guanine. This pairing forms the rungs of the ladder, while the sides are made of sugar and phosphate molecules. But the structure is only part of the story. What makes DNA remarkable is the sequence of its bases. Like letters forming words, these sequences carry specific instructions, not just for making proteins, but for when and where to make them, and in what quantities. These sections of DNA are known as genes. Each gene contains the code for a specific protein, a molecule that performs a task or builds a structure within the cell. But DNA itself does not build proteins. It provides the instructions, and another molecule carries them out. That molecule is RNA. In a process known as transcription, a section of DNA is copied into a single-stranded messenger RNA, or mRNA. This RNA strand then exits the nucleus through a nuclear pore and travels to a ribosome, where translation occurs, the process of building the protein according to the RNA's instructions. This separation of roles, DNA storing the instructions and RNA delivering them, adds a layer of regulation. The nucleus becomes not just a container of information, but a site of decision-making. Only certain genes are copied at any given time, depending on the needs of the cell. This careful control is what allows a skin cell to differ from a liver cell, even though both contain the same DNA. At the center of the nucleus, we find the nucleolus, a dense, dark region that serves a vital function, assembling ribosomes. These are the protein-making factories of the cell, and they themselves are made of RNA and protein. Once partially assembled in the nucleolus, ribosomes are sent out to the cytoplasm, where they complete their structure and begin their work. Despite its stillness, the nucleus is a site of ceaseless activity. DNA is coiled and uncoiled. RNA is transcribed and edited. Signals from the outside world, hormones, nutrients, stressors, are translated into responses at the genetic level. The cell adjusts, adapts, and continues. Within each human cell, there are 46 chromosomes, arranged in 23 pairs. These contain approximately 20, 0 to 25, 0 genes, spread across about 3 billion base pairs. Yet even with this vast amount of information, the cell uses only a portion at any given time. Some genes are active, constantly housekeeping genes that keep the cell running. Others are switched on or off, depending on the cell's function, its stage of development, or the conditions it encounters. This selective expression gives rise to cellular identity. A neuron expresses a different suite of genes than a muscle cell. The DNA is the same, but the expression, the song it sings, is different. This regulation is not random. It is guided by transcription factors, proteins that bind to specific sequences of DNA and determine which genes are read. It is also shaped by epigenetic modifications, chemical tags that can tighten or loosen DNA's coiling, making it more or less accessible. Such modifications do not change the genetic code itself, but they influence how the code is used. They are like bookmarks or notes in the margin, a way for the cell to remember which pages it needs and which ones it can leave untouched. Even DNA repair is quietly underway. Every day your DNA experiences damage from sunlight, from chemicals, from ordinary cellular processes. But the nucleus houses repair enzymes that scan for errors, cut out damaged sections, and stitch the strands back together. In this way, the nucleus is both guardian and guide, preserving the genetic code and ensuring its careful use. Perhaps most humbling of all is the continuity of this material. The DNA in your cells was copied from that of your parents, which was copied from theirs, and so on, stretching back through time. It is not just a molecular instruction set. It is a thread of history unbroken, flowing from generation to generation, cell to cell. Every cell in your body carries a complete copy of your genome. Even though most of that DNA remains unread in any given moment, it is always there, a quiet readiness, a deep potential. And so, as we reflect on the nucleus and the DNA it holds, we see not just function, but legacy. Within this soft, membranous chamber lies a language spoken by all life, written in molecules, passed through time, and carried within you now in the gentle, silent heart of every cell. Part 8. Organelles in Harmony. As we return once more to the soft interior of the eukaryotic cell, we find ourselves surrounded not by chaos, but by quiet coordination. This is a space of cooperation, a place where distinct compartments, or organelles, each carry out their roles in a harmonious flow. There is no sound here, no commanding voice, and yet every part knows what to do and when to do it. The cell is a symphony of function, and its organelles are the instruments. Let us begin again with the mitochondrion, the organelle often called the powerhouse of the cell. These bean-shaped structures are the sites of cellular respiration, a series of chemical reactions that transform nutrients into usable energy. That energy takes the form of ATP, or adenosine triphosphate, the molecule that fuels most of the cell's work. Mitochondria have two membranes, a smooth outer layer and a highly folded inner layer. The folds, called cristae, increase the surface area available for energy production. Within these folds, glucose and oxygen are combined in a gentle process that releases energy, water, and carbon dioxide. What makes mitochondria even more remarkable is that they carry their own DNA separate from the cell's nuclear DNA and can replicate independently. This supports the idea that they were once free-living organisms, gradually incorporated into early cells in a relationship that became permanent. In plant cells, a similar story unfolds in the chloroplasts. These green organelles perform photosynthesis, using sunlight, water, and carbon dioxide to produce sugars. Like mitochondria, chloroplasts have their own DNA and multiple membranes. Their internal structure includes stacks of disc-like membranes called thylakoids, where chlorophyll captures light. The sugar produced here becomes food not only for the plant, but for every organism that feeds on it directly or indirectly. Both mitochondria and chloroplasts represent the cell's energy systems, one using food, the other using light. They do not compete, but complement each other, depending on the type of cell and its role in the organism. Close by, we find the endoplasmic reticulum, or ER, an extensive network of membranes that weaves through the cytoplasm, like a quiet transport system. The ER exists in two forms, rough and smooth. The rough ER is covered with ribosomes, giving it a textured appearance. These ribosomes assemble proteins based on instructions carried by messenger RNA. As the proteins are built, they enter the ER's channels, where they may be folded, modified, or prepared for export. The smooth ER, on the other hand, lacks ribosomes and focuses on different tasks. It synthesizes lipids, the fats and oils that form membranes and hormones. It also plays a role in detoxifying harmful substances and storing calcium ions. In liver cells, for instance, the smooth ER helps break down medications and alcohol, quietly protecting the body from toxins. From the ER, many proteins and lipids are sent to the Golgi apparatus, or Golgi body, a stack of flattened, membrane-bound sacs. The Golgi modifies, sorts, and packages these molecules, placing them into small vesicles for delivery. Some vesicles remain inside the cell, others fuse with the membrane to release their contents outside. If the ER is the factory, the Golgi is the post office, labeling and routing each product with precision. Elsewhere in the cell, small, spherical lysosomes serve as the recycling centers. They contain powerful enzymes that break down damaged organelles, old proteins, and engulfed particles. These enzymes function best in acidic conditions, and the lysosome maintains this environment carefully, keeping its contents separate from the rest of the cell. Lysosomes play a key role in cellular housekeeping. Without them, waste would accumulate, and vital space would be lost. In many ways, they are silent caretakers, dissolving the old to make room for the new. Another structure, found primarily in animal cells, is the peroxisome. These small organelles break down fatty acids and detoxify substances like hydrogen peroxide, converting it into water and oxygen. Like lysosomes, they protect the cell by safely containing their reactions. In plant cells, we often find a large central vacuole, a storage chamber for water, nutrients, pigments, and waste. This vacuole also helps maintain the cell's shape by creating turgor pressure, pushing outward against the cell wall. It is a still reservoir, holding the materials that the cell may need later or that it must isolate for safety. Smaller vacuoles are found in animal cells as well, where they serve roles in storage and transport. Connecting all these parts is the cytoskeleton, a network of protein filaments and tubules that crisscross the cytoplasm. The cytoskeleton provides structure, much like the frame of a house, but it is not static. It assembles and disassembles as needed, guiding the movement of organelles, shaping the cell, and enabling motion. There are three main types of fibers in the cytoskeleton. Microfilaments made of actin support the cell's shape and assist with movement. Intermediate filaments provide mechanical strength. Microtubules, hollow tubes made of tubulin, form tracks for moving vesicles and help separate chromosomes during cell division. In animal cells, two small structures called centrioles are found near the nucleus. These help organize the spindle fibers that pull chromosomes apart during mitosis. Together with surrounding material, they form the centrosome, a key player in cell division. Floating freely in the cytoplasm, or attached to the ER, are the ribosomes' tiny complexes of RNA and protein. Though not bound by a membrane, ribosomes are essential organelles in their own right. They read the genetic code delivered by RNA and build proteins, one amino acid at a time, folding them into the shapes that determine their function. And finally, enveloping it all is the cell membrane, a flexible boundary that holds the entire system together. It is more than a barrier. It is an active participant, constantly adjusting, sensing, and responding. It is the surface through which the cell communicates with its world. Each organelle, from the largest nucleus to the smallest vesicle, plays a part in the life of the cell. None operates in isolation. Their functions are linked, their timings coordinated, their roles complementary. Together, they sustain the quiet miracle of a living cell. As you imagine this world, invisible, soft, and endlessly organized picture, a living city at rest. Its structures glow softly, its processes unfold without haste, its boundaries shift and reform with fluid grace. The cell is not a machine, it is a living harmony. And within you, trillions of these cells hum in concert, each one following the same quiet choreography, organelles in harmony, sustaining the body in stillness. Part 9, the cell membrane. As we continue our calm journey through the inner landscape of the cell, we now come to the boundary that defines its very shape, the cell membrane. It is not merely a wall, nor a simple wrapper. It is a living, flexible, highly selective gateway, a structure so thin it cannot be seen without magnification, and yet so essential that life could not proceed without it. The cell membrane, also known as the plasma membrane, wraps around every cell like a quiet, responsive skin. It separates the inner world from the external environment, maintaining conditions suitable for life. But unlike a rigid shell, the membrane is soft, semi-permeable, and remarkably active. Let us ease into its structure. The core of the membrane is made from a phospholipid bilayer, two layers of fat-like molecules arranged tail to tail. Each phospholipid molecule has a hydrophilic head that is attracted to water and hydrophobic tails that repel it. In the bilayer, the heads face outward toward the watery environments both inside and outside the cell, while the tails point inward, away from water, forming a barrier. This arrangement is not static. The phospholipids can drift sideways, flexing and shifting like a liquid sheet. This fluidity allows the membrane to change shape, repair itself, and engulf substances in a process called endocytosis. Floating within and between these lipid molecules are proteins, some that span the entire membrane, others that rest on one side. These proteins serve many functions. Some act as transport channels, helping substances cross the membrane. Others are receptors, receiving signals from the environment and triggering internal responses. Still others are anchors, connecting the cell's internal structure to its exterior surroundings. Together, the lipids and proteins form a mosaic, a soft patchwork of movement and function, known as the fluid mosaic model. Also embedded in the membrane are cholesterol molecules in animal cells. These act as stabilizers. When temperatures drop, cholesterol prevents the membrane from becoming too rigid. When temperatures rise, it helps keep the membrane from becoming too fluid. In this way, cholesterol contributes to the quiet adaptability of the membrane. The outer surface of the membrane may also be decorated with carbohydrate chains attached to lipids or proteins. These structures help the cell recognize and interact with other cells. Like gentle name tags, they allow immune cells to identify friend from foe and help tissues organize during growth and repair. One of the membrane's most important features is its selective permeability, the ability to allow some substances through while blocking others. This function is vital for maintaining the internal environment of the cell, a state known as homeostasis. Small nonpolar molecules like oxygen and carbon dioxide can slip through the lipid bilayer easily, moving from areas of higher concentration to areas of lower concentration. This process, requiring no energy, is known as simple diffusion. Water, though polar, can also move across the membrane in a special way. It travels through proteins called aquaporins, using a type of passive transport called osmosis. Water flows toward areas of higher solute concentration, balancing the internal and external conditions gently, without push or command. For larger or charged molecules, the membrane uses facilitated diffusion. Here, specific transport proteins create channels or carriers, allowing substances like glucose or ions to move along their concentration gradient again, without energy expenditure. But sometimes the cell must move substances against the natural flow from areas of low concentration to high. This requires active transport, which uses energy in the form of ATP. Proteins known as pumps perform this task. A well-known example is the sodium-potassium pump, which moves sodium ions out of the cell and potassium ions in, maintaining the electrical balance essential for nerve function and muscle movement. Beyond transport, the membrane plays a key role in communication. Cells receive signals from hormones, neurotransmitters, or environmental cues. These signals bind to receptor proteins on the membrane, triggering cascades of internal changes. The cell may respond by altering its shape, changing its behavior, or even beginning a new process like division or repair. This ability to sense and respond makes the cell membrane not just a boundary, but a sensor, the first point of contact between a cell and its world. The membrane is also involved in the cell's ability to engulf and release larger particles. In endocytosis, the membrane folds inward, wrapping around a particle and drawing it inside. In exocytosis, vesicles inside the cell fuse with the membrane, releasing their contents into the surrounding space. These processes allow cells to eat, drink, and communicate not with words, but with movement and molecules. In multicellular organisms, membranes help cells stick together and form tissues. Adhesion proteins on the surface allow cells to connect, creating sheets, layers, and networks. In some tissues, gap junctions form channels that directly link neighboring cells, allowing ions and small molecules to pass through. In plant cells, a similar structure called the plasmodesma bridges cell walls, creating a silent conversation between adjacent cells. And yet, for all its activity, the cell membrane remains unseen by the naked eye. It is a boundary without bulk, a skin without stiffness. Its presence is felt in the quiet regulation of every breath you take, every heartbeat, every sensation. It holds the internal steady, protects the delicate, and welcomes the needed. When we think of intelligence, we often picture the brain, but at a smaller scale, the membrane exhibits a kind of cellular wisdom, balancing inputs and outputs, maintaining shape, sensing surroundings, and adapting to change. It is, in essence, the voice of the cell at the edge where inner life meets the outside world. So as you rest, remember this: every cell in your body, from fingertip to spine, is wrapped in a membrane just like this, fluid, thin, aware, and within that quiet boundary, the work of life unfolds. Part 10. Energy in the cell. All life requires energy. Without it, cells could not grow, divide, or maintain their inner balance. Muscles would not contract, thoughts would not form, and even the smallest action like the blink of an eye would never begin. Yet this essential force does not come in bursts or lightning strikes. Instead, it flows through living things quietly, invisibly, moment by moment. In the cell, energy is managed with extraordinary care. It is not hoarded nor wasted. It is transformed, transferred, and used always in gentle moderation, always with balance. Let us begin with the cell's main energy currency, adenosine triphosphate, or ATP. ATP is a small molecule, but its role is vast. It stores energy in the form of chemical bonds, specifically the high-energy bonds between its three phosphate groups. When the cell needs energy, it breaks one of these bonds, converting ATP into ADP, or adenosine diphosphate. This release is controlled and subtle, like the strike of a match, enough to power a reaction, but never more than needed. The making of ATP is central to cellular life. The most efficient method for producing it is called cellular respiration, a series of chemical reactions that convert glucose, a simple sugar, into usable energy. Cellular respiration takes place mainly in the mitochondria, often described as the cell's power plants. These organelles contain the enzymes and spaces needed to perform this complex task. The process begins in the cytoplasm, where glucose is broken down into smaller molecules through a pathway known as glycolysis. This step does not require oxygen and produces a small amount of ATP, along with molecules that carry energy in other forms, NADH and pyruvate. The pyruvate then travels into the mitochondria, where the process continues. In the Krebs cycle, also called the citric acid cycle, more energy-carrying molecules are created. Finally, in the electron transport chain, electrons from these molecules are passed along a series of protein complexes embedded in the mitochondrial membrane. As electrons move through the chain, they release energy, which is used to pump hydrogen ions across the membrane. This creates a gentle gradient, a difference in concentration, like water behind a dam. When the ions flow back through a special enzyme called ATP synthase, the stored energy is used to convert ADP into ATP. This process, known as oxidative phosphorylation, produces the majority of the cell's ATP, often over 30 molecules from a single glucose molecule. Oxygen, quietly inhaled with each breath, acts as the final electron acceptor in this chain, combining with hydrogen to form water. The cell's use of oxygen in this way is why we breathe. It is why lungs draw in air and blood delivers it to every tissue. Each molecule of oxygen ends its journey inside a mitochondrion, accepting electrons and allowing energy to be made. But what happens when oxygen is scarce? Cells have backup systems slower, less efficient, but effective in short bursts. In the absence of oxygen, cells can perform anaerobic respiration or fermentation. In human muscle cells, this leads to the production of lactic acid, a substance that can cause a gentle ache during intense activity. Though less ATP is made, the cell continues to function, drawing energy where it can. In yeast and certain bacteria, fermentation produces alcohol and carbon dioxide instead. This quiet chemistry is the basis of bread rising and beer brewing processes that rely on microbial cells doing what they have done for millions of years, transforming sugar into energy, even in the dark. Beyond glucose, cells can use other fuels. Fats are broken down into smaller units that enter the respiration pathway. Proteins, too, can be used, though this is less common. The body prefers to spare proteins for their structural and functional roles. The regulation of energy within the cell is finely tuned. Enzymes act as gatekeepers, turning reactions on or off as needed. Feedback loops prevent waste. Signals from hormones such as insulin or adrenaline adjust the pace of energy use based on the body's needs. It is a quiet, ongoing calculation. How much energy is available, how much is required, and where it must go. Energy is not only about movement or heat, it is used for active transport, where molecules are moved against gradients across membranes. It fuels the synthesis of macromolecules, proteins, nucleic acids, and lipids. It powers the beating of cilia, the contraction of muscle fibers, the replication of DNA, and the repair of damage. Even the simple maintenance of a stable internal environment, the right balance of ions, pH, and temperature, relies on constant energy use. In plants and algae, energy enters in a different way. Through photosynthesis, light is captured by chloroplasts and transformed into sugar. This sugar, in turn, is used in respiration, the two processes forming a gentle loop between light and life, air and energy. Chloroplasts and mitochondria work in tandem. One captures energy from the sun, the other releases it for use. One consumes carbon dioxide and gives off oxygen. The other consumes oxygen and gives off carbon dioxide. In this way, photosynthesis and respiration mirror one another, a calm, planetary rhythm that sustains all ecosystems. In every living cell, energy flows. It is never created or destroyed, but constantly transformed from light to sugar, from sugar to ATP, from ATP to motion, repair, or thought. It is passed along quietly, a molecular current within the sea of life. This flow of energy connects all living things, from a blade of grass absorbing sunlight to a hummingbird's rapid wing beat, from the stretching of muscle to the growth of a seedling. Every act begins with energy made available by a cell. And so, as you rest, your own cells continue their work. In the stillness of mitochondria, glucose is transformed. Tiny molecular machines turn, gradients shift, and ATP is passed hand to hand, a silent engine keeping you alive. It is not dramatic, it is not loud, but it is unceasing, and it is enough. Part 11, Protein Synthesis. In the gentle workings of the cell, few processes are as constant or as quietly vital as the making of proteins. These complex molecules form the tools, structures, and signals that sustain every aspect of life. From the pigments in your eyes to the enzymes that digest your food, proteins are everywhere. They build, they carry, they regulate, they respond, and each one begins the same way, with a line of code, written softly in the language of DNA. This entire process, known as protein synthesis, unfolds in two main stages: transcription and translation. Though the terms may sound mechanical, the process is graceful, precise, and deeply elegant. Let us begin in the nucleus, where the instructions for making proteins are stored as gene segments of DNA. Each gene holds the specific sequence of bases, adenine, thymine, cytosine, and guanine, that spell out how to build one particular protein. But DNA itself does not leave the nucleus. To carry the instructions into the wider cell, a messenger molecule must be made. This is the role of RNA specifically, messenger RNA, or mRNA. In the first stage, transcription, the cell gently opens a section of DNA. Specialized enzymes, called RNA polymerases, move along one strand of the DNA, reading its bases, and building a complementary strand of mRNA. Where the DNA has adenine, the RNA adds uracil, a soft substitute for thymine. The rest of the bases pair as usual, cytosine with guanine, and so on. As this single-stranded RNA is formed, It carries with it the exact instructions to build the target protein copied faithfully from the genetic code. When the mRNA strand is complete, it detaches and leaves the nucleus through a nuclear pore, entering the cytoplasm, where the second stage begins. Translation. Here, the mRNA encounters a ribosome, the molecular machine that will build the protein. Ribosomes may float freely in the cytoplasm or be attached to the rough endoplasmic reticulum, giving it its textured appearance. The ribosome reads the mRNA 3 bases at a time. Each set of 3 is called a codon, and each codon corresponds to one amino acid, the building blocks of proteins. To bring these amino acids into place, the cell uses another kind of RNA transfer, RNA, or tRNA. Each tRNA molecule carries a specific amino acid on one end and has a complementary anticodon on the other, a code that matches with the mRNA's codon. As the ribosome moves along the mRNA strand, tRNAs arrive, one by one, matching their anticodons to the codons and placing the correct amino acid in position. The ribosome helps link the amino acids together with peptide bonds, creating a long, growing chain. This chain is known as a polypeptide, and as it lengthens, it begins to fold, not randomly, but with intention. Proteins do not work as straight lines. They fold into three-dimensional shapes, held together by subtle forces: hydrogen bonds, ionic interactions, hydrophobic pockets, and sometimes even covalent bridges. This shape, this conformation, determines the protein's function. Some proteins act as enzymes, speeding up reactions. Others become receptors, hormones, antibodies, or structural components like keratin or collagen. Their diversity is nearly endless, but their origins are always the same: DNA, RNA, protein. Once the protein is built and folded, it is sometimes modified. Further sugars may be added, sections trimmed, or parts combined with other proteins. These finishing touches often occur in the Golgi apparatus, where the protein is packaged into a vesicle and sent to its final destination, the cell membrane, a specific organelle, or out into the bloodstream. This entire journey from gene to final product is directed by the cell without noise or oversight. No thought guides it, no eyes watch. And yet, it is remarkably accurate. Cells can produce thousands of different proteins, some in vast quantities, others in tiny amounts. They regulate protein synthesis through gene expression, turning genes on or off, depending on need, environment, or stage of development. This flexibility allows the same DNA to give rise to many cell types. A neuron and a liver cell have identical genetic material, yet each expresses a unique set of proteins. This selective reading of the genome is what creates specialization, what allows your body to be both unified and diverse. And when a protein is no longer needed, the cell quietly breaks it down, recycles its parts, and clears the space for something new. There is no waste, only renewal. This quiet efficiency is why protein synthesis is so central. Without it, the cell would be lifeless, unable to adapt, repair, grow, or communicate. Proteins are not just tools. They are the agents of life itself. Even in sleep, your cells are building proteins. Right now, in your muscles, in your brain, in your bones, ribosomes are clicking along strands of mRNA, piecing together amino acids like beads on a string. The hum of synthesis never truly stops. It is a continuous unfolding of code into form language made flesh. And within that process, there is something deeply peaceful, a rhythm, a pattern, a quiet purposefulness. A gene is read, a message is sent, a chain is built, a shape is formed, and a new part of life begins its work, all without sound, all within you. Part 12. Cellular communication. Within the quiet architecture of living tissues, a hidden conversation unfolds not in words or symbols, but in the language of molecules and movement. This is cellular communication, the way individual cells sense their surroundings, share information, and coordinate their responses. Though cells lack voices, ears, or conscious awareness, they speak a language more ancient than any spoken tongue. They send signals, receive messages, and act together with remarkable unity. From a distance, tissues and organs may appear as fixed structures, but inside, a network of silent exchange connects every cell. Let us begin with the simplest form: chemical signaling. Cells release molecules called signal molecules or ligands into their environment. These molecules drift through fluids or across membranes until they encounter a receptor, a protein structure, embedded in or on the surface of another cell. When a ligand binds to its receptor, the shape of the receptor changes, and with this subtle change, a cascade of internal responses begins. There are several ways this process can occur, depending on how far the signal must travel. In autocrine signaling, a cell releases a signal that binds to receptors on its own surface. It listens to its own message, a way of reinforcing or regulating its own behavior. In paracrine signaling, the signal affects nearby cells. This is common during development, when cells guide one another's growth and arrangement. In tissues, immune cells use this form to coordinate their defense, sending gentle alerts to those nearby. When signals must travel further, endocrine signaling is used. Here, specialized cells release hormones into the bloodstream, where they are carried to distant cells throughout the body. Insulin, for example, is released by cells in the pancreas but affects cells in muscles, liver, and fat, adjusting how they absorb and store sugar. There is also direct contact where neighboring cells communicate through shared channels. In animal cells, gap junctions form small pores that link the interiors of adjacent cells, allowing ions and small molecules to flow from one to the next. In plant cells, similar connections are called plasmodesmata, which bridge cell walls and allow exchange of nutrients and messages. Once a signal reaches its target, it must be understood. The receptor that receives the signal translates it into an internal event, a process called signal transduction. This often involves a series of steps where one molecule activates another, and so on, like a chain of gentle handshakes. These steps may lead to changes in the cell's metabolism, movement, shape, or even gene expression. Sometimes the response is quick, like a muscle contracting. Other times it is slow, like a cell beginning to divide, or shifting its role within a developing tissue. Not all signals come from other cells. Cells also respond to their environment, to light, temperature, touch, and the concentration of ions or nutrients. Some cells in your eye, for instance, contain proteins that change shape when struck by light, beginning the cascade that leads to vision. In your inner ear, sensory cells respond to vibrations, transforming sound into electrical impulses. In your skin, gentle pressure is converted into signals that reach the brain, interpreted as touch. The immune system, too, is guided by cellular communication. When a virus enters the body, infected cells release signaling molecules known as cytokines, which alert immune cells. White blood cells respond by migrating to the site of infection, guided by gradients of these molecular signals. In every tissue, communication ensures cooperation. Cells signal when they need nutrients. They signal when they are damaged. They signal when it is time to grow, to divide, or to cease. Even in the process of death, apoptosis, or programmed cell death, cells release signals to neighbors, ensuring that cleanup occurs quietly, without disruption. Mistakes in communication can lead to disease. If signals are too strong or too weak, or if receptors malfunction, the results can be profound. Uncontrolled cell division may lead to cancer. Faulty insulin signaling leads to diabetes. Misinterpreted immune signals may cause inflammation or autoimmunity. These disruptions remind us how delicate and how vital the communication between cells truly is. Yet most of the time, it works beautifully. Even now, messages pass through your body in silent harmony. Heart cells contract in unison. Skin cells replace themselves at the surface. Neurons pass thoughts and memories as electrical signals and chemical whispers. In your brain, billions of neurons communicate across narrow gaps called synapses. Here, an electrical impulse reaching the end of one neuron causes it to release neurotransmitters, chemical messengers, into the synaptic cleft. These molecules drift across and bind to receptors on the next neuron, passing the message forward. This happens with extraordinary speed and precision, not just in conscious thought, but in balance, breathing, heartbeat, and dreams. The quiet signals of the nervous system guide everything from motion to mood. Beneath this neural activity, other cells continue their conversations. Liver cells monitor blood chemistry. Bone cells respond to weight and pressure. Blood cells signal where they are needed and when their work is done. Communication is not constant chatter. It is quiet when needed, urgent when called for, and always purposeful. A cell does not speak without reason. It listens as much as it speaks. Each signal is like a drop of ink in water spreading gently, reaching its target, changing the shape of the cell's understanding. And though these signals are invisible, they form the language of life itself. So tonight, as you lie still and drift into sleep, the cells within you are anything but silent. They are signaling, sensing, and responding, carrying out an unbroken conversation that began when your body first formed, and will continue until its very last breath. There is no noise, only connection. A quiet world, alive with meaning. Part 13, Cell Division. Mitosis. Every living organism begins as a single cell. From that quiet origin, life unfolds through growth, renewal, and reproduction. The way this happens, at its most essential level, is through the process of cell division, a calm, deliberate act in which one cell becomes two. This process is not rushed. It is not erratic. It is carefully timed, meticulously executed, and repeated billions of times within the human body each day. The type of division we will now explore is called mitosis, the process by which a single cell creates two genetically identical daughter cells. This is how tissues grow, how wounds heal, and how cells replace those that are old, damaged, or worn. Let us begin with the quiet phase that sets the stage, interphase. Though not technically part of mitosis, interphase is where a cell spends most of its life. During this time, the cell grows, performs its regular functions, and prepares for division. Interphase itself has 3 stages. Gon phase, where the cell grows and synthesizes proteins. S phase, where the DNA is replicated, ensuring each daughter cell will receive a complete set of genetic instructions. 2 phase, where the cell continues to grow and checks for any errors in the duplicated DNA. At the end of interphase, the cell is ready. Now begins mitosis, a process not of violence, but of separation, precision, and renewal. Mitosis is classically divided into 4 main stages: prophase, metaphase, anaphase, and telophase, followed by the final act of cytokinesis. In prophase, the chromatin, the relaxed, thread-like form of DNA, begins to coil and condense into visible chromosomes. Each chromosome now consists of 2 identical sister chromatids, joined at a central point called the centromere. At the same time, the nuclear envelope begins to break down, and structures known as centrioles move to opposite ends of the cell. Between them, a network of protein fibers begins to form the mitotic spindle. These fibers will guide the chromosomes like silent shepherds. In metaphase, the chromosomes line up along the center of the cell, forming a straight, silent row at what is called the metaphase plate. The spindle fibers attach to the centromeres, ensuring each chromatid is connected to an opposite pole. This alignment is not just orderly, it is crucial. If the chromosomes do not line up correctly, the cell will pause, delaying division until everything is secure. The cell checks and rechecks, ensuring nothing is rushed. Then comes anaphase. In this stage, the centromeres split and the sister chromatids are gently pulled apart. Now considered individual chromosomes, they are drawn to opposite ends of the cell, guided by the shortening spindle fibers. This is a moment of separation, but not of destruction. It is a mirror unfolding. Each side now holds a complete set of chromosomes, identical to the original. In telophase, the cell begins to rebuild. New nuclear envelopes form around each set of chromosomes. The spindle fibers disassemble. The chromosomes begin to uncoil, returning to their relaxed, thread-like form. Two distinct nuclei are now visible, each a quiet copy of the other. Mitosis is complete, but the cell is not yet fully divided. The final step is cytokinesis, the division of the cytoplasm. In animal cells, a ring of protein fibers tightens around the center of the cell, creating a cleavage furrow that deepens until the cell is pinched in two. In plant cells, which have rigid walls, a cell plate forms instead, built from vesicles that merge to form a new dividing wall between the two daughter cells. And just like that, two new cells are born. Each one is fully functional, fully equipped, and genetically identical to the cell that gave rise to them. They begin their own interphases, growing, working, and one day perhaps dividing again. The purpose of mitosis is not only reproduction but continuity. It ensures that the body's tissues remain intact and renewed. Skin cells are constantly replaced. Blood cells wear out and are replenished. Even the lining of your stomach, subjected to acids and enzymes, is quietly rebuilt through mitosis. It is a peaceful cycle of life within the body, no fanfare, no rush, just one cell becoming two. Errors in this process are rare, but when they occur, they can lead to problems. If a cell divides uncontrollably, ignoring the body's signals, a tumor may form. If chromosomes are distributed unevenly, genetic disorders may arise. And yet, for the most part, cells divide with astonishing accuracy, checking their work, correcting mistakes, and preserving the pattern of life. There is also another type of cell division, meiosis, used to create gametes, such as sperm and egg cells. Unlike mitosis, meiosis reduces the number of chromosomes by half, ensuring that when two gametes fuse, the resulting embryo has the correct number. But that is a story for another night. Tonight, we rest with mitosis, the quiet rhythm of growth and renewal. As you lie still, billions of your cells are preparing to divide, or are already doing so. Skin cells, intestinal cells, cells deep within your bones. Each one repeats the same ancient steps: prophase, metaphase, anaphase, telophase, as if reciting a lullaby passed down through time. Each new cell holds the full blueprint of life. Each one carries the same silent purpose: to live, to function, and to continue the work of the whole. Mitosis is not dramatic. It is not loud, but it is constant. It is faithful, and it is life's way of saying without words that the work will go on. Part 14. Specialized cells. As we journey deeper into the quiet intricacies of biology, we now encounter a beautiful truth. Not all cells are the same. Though they may begin with identical instructions, the same DNA, the same blueprint, many cells grow into different forms, each uniquely shaped by its role in the body. These are the specialized cells, the quiet craftsmen of life. each performing a task both essential and refined. This process, known as cellular differentiation, allows a single fertilized egg, one cell, to give rise to the vast complexity of the human body. Over time, and through careful coordination, cells take on distinct identities. Some become long and lean, others flatten, branch, or fill with pigment. Their forms mirror their functions. Let us gently explore some of these cell types, appreciating the silent expertise of each. Begin with the nerve cell, or neuron. Long and slender, with delicate arms reaching outward, neurons are the messengers of the nervous system. Their shape allows them to send signals over great distances. A neuron receives information through its dendrites, processes it in the cell body, and transmits it along a long axon, sometimes stretching over a meter in length, to reach other cells. These electrical impulses, quiet as whispers, are translated into chemical signals at the synapse, allowing thoughts, sensations, and movements to unfold. Your ability to read, dream, and remember is made possible by the tireless work of neurons, each one precisely shaped for its task. Now consider the muscle cell. Also known as muscle fibers, these cells are long and contractile. Their interior is packed with filaments of actin and myosin proteins that slide past one another to produce movement. In skeletal muscles, cells are bundled into fibers, allowing limbs to move and posture to be maintained. In the heart, cardiac muscle cells beat rhythmically, contracting in perfect synchrony. And in the walls of internal organs, smooth muscle cells move food, blood, or air gently and continuously. Each type of muscle cell is suited to its environment, shaped for strength, endurance, or rhythm. Their forms are not accidental. They are sculpted by need. Floating through your bloodstream are red blood cells, small, round, and concave. They lack a nucleus and most organelles, making room for hemoglobin, the protein that binds and transports oxygen. Their shape allows them to bend and squeeze through narrow capillaries, delivering oxygen to every corner of the body, then returning with carbon dioxide for release. These cells are silent carriers, moving with the pulse of the heart, never stopping, never speaking, just delivering life. Also in the blood are white blood cells, or leukocytes, part of the immune system. These cells come in many forms. Some engulf invaders, others produce antibodies, and still others coordinate responses. They move through tissues, scanning for signs of trouble, and respond with precision. Though fewer in number than red blood cells, their presence is vital. They are defenders, helpers, and memory keepers, ensuring the body knows friend from foe. Let us not forget the epithelial cells, those that form the linings and coverings of the body. From the skin's surface to the lining of the lungs, the digestive tract, and the blood vessels, epithelial cells create barriers. They protect, absorb, secrete, and sense. In the lungs, they are thin and flat, allowing gases to pass. In the intestine, they are tall and covered in microvilli, increasing surface area for absorption. Despite their differences, all epithelial cells are closely joined, forming sheets that shield and separate. Their arrangement is quiet and orderly, like tiles placed by a careful hand. Within bones, we find osteocytes, mature bone cells embedded in hard mineral. These cells monitor the bone's condition and direct remodeling. Osteoblasts build new bone, while osteoclasts dissolve old material. The result is a structure that is strong yet adaptable, reshaping itself over time in response to stress. In connective tissues, fibroblasts produce collagen and other fibers, creating the supportive framework of the body. In fat tissue, adipocytes store energy and insulate. In the reproductive system, gametes, sperm, and egg cells are highly specialized for their role in passing on life. Each of these cell types arises from a less specialized form, often from a stem cell. Stem cells are the body's unspecialized reservoir. In early development, they can become any type of cell, a property known as pluripotency. Later in life, some stem cells remain in tissues, ready to divide and replace lost or damaged cells. These adult stem cells are more limited in scope, but no less important. In the bone marrow, for example, stem cells constantly replenish the blood. What guides this differentiation? Signals both internal and external shape a cell's fate. Chemical gradients, contact with neighbors, and patterns of gene expression determine which genes are active and which are silent. Though every cell contains the full genome, only a subset is used, forming each cell's identity. This selective expression creates the beautiful diversity of tissues, the layers of skin, the spirals of muscle, the networks of neurons. All emerge from a single cell, dividing and differentiating with calm precision. And even in specialization, there is collaboration. No cell works alone. Neurons send signals to muscles. Blood delivers oxygen to skin. The immune system monitors every tissue. This quiet cooperation forms the fabric of the body, a living mosaic of specialized cells, each doing its part. Right now, within you, these cells continue their quiet work. Some are transmitting signals, others are contracting. Some are filtering, absorbing, patrolling, or supporting. They are shaped by purpose, formed by history, and sustained by rhythm. And though they differ in form, they remain united in origin. Each cell traces back to the same beginning, one fertilized egg, one cell that divided, again and again, creating complexity from simplicity. Specialization is the flowering of life, a way for cells to do more, together, than any could do alone. So as you rest, your body hums with harmony, a quiet chorus of specialized cells, each fulfilling its task, each part of something greater than itself. Part 15, Stem Cells and Development. As we continue our gentle exploration of the living cell, we now turn our attention to something both foundational and full of possibility, the stem cell. These are the unspecialized cells, the quiet origin from which all other cell types emerge. They are the starting point for every heartbeat, every breath, every layer of skin, and sweep of bone. Stem cells represent life's potential in its calmest, purest form, and in the soft language of biology, they speak of beginnings. Every organism, including the human body, begins as a single cell, the zygote formed when a sperm and an egg unite. From that one cell, an entire human being will unfold. But the zygote is not a finished product. It is a promise, and it keeps that promise by dividing, again and again. With each division, cells begin to make choices. Some of these early cells remain flexible, able to become any type of cell in the body. These are known as pluripotent stem cells. In the very earliest stages of embryonic development, these cells divide and multiply, forming a structure known as the blastocyst, a hollow ball of cells that contains the inner cell mass, where pluripotent stem cells are concentrated. From these stem cells come every tissue, every organ, every specialized cell in the body, muscle, nerve, skin, bone, and more. This transformation, from unspecialized to specialized, is known as differentiation. Differentiation is guided by gentle cues, chemical signals, gene expression patterns, and spatial organization. Depending on their position in the embryo and the signals they receive, stem cells begin to express specific genes, folding into new identities. The result is a quiet cascade of decisions, each leading cells closer to their final forms. As development progresses, pluripotent stem cells give rise to more limited, multipotent stem cells, those that can still become many types of cells, but within a certain family. For example, hematopoietic stem cells in the bone marrow can become any type of blood cell, but not a nerve or muscle cell. Even in adulthood, some stem cells remain. These are called adult stem cells, or somatic stem cells. They are found in various tissues, skin, blood, intestines, where they help maintain and repair the body. In the lining of the gut, for instance, stem cells divide daily to replace cells lost during digestion. In the skin, they replenish the outer layers as they wear away. And in the bone marrow, they continually generate red and white blood cells, keeping the body nourished and protected. There is also growing interest in induced pluripotent stem cells, or iPS cells, ordinary adult cells that scientists reprogram to return to a pluripotent state. By gently altering gene expression, these cells regain the flexibility of embryonic stem cells, opening new paths for research and therapy. In all their forms, stem cells are defined by two quiet powers. 1. Self-renewal, the ability to divide and produce more stem cells. 2. Potency, the potential to differentiate into specialized cells. These two traits allow tissues to grow, heal, and adapt. When you scrape your knee, stem cells in the skin respond. When your blood supply is low, bone marrow stem cells increase their output. The body does not panic. It simply begins again. In developmental biology, the journey from stem cell to specialized cell is called cell lineage, a map of descent, like a family tree. Each branch leads to a specific type of cell, each fork a decision made silently, deep within the embryo. As organs form the heart, the lungs, the eyes, stem cells provide the raw material. But more than that, they provide timing, shape, and structure. The heart begins as a simple tube, the brain as a folded sheet. Through proliferation and differentiation, the embryo takes form, sculpted not by hand, but by the behaviors of its cells. And even long after birth, this development continues. In children, stem cells drive growth. In adults, they maintain. In both, they respond to need, quietly, unfailingly. In medicine, stem cells hold great promise. Already, stem cell transplants are used to treat blood cancers. One's own stem cells can help rebuild damaged tissues. And in the future, stem cells may be used to generate organs for transplantation, to repair spinal injuries, or to reverse degenerative diseases. But even without intervention, the body's own stem cells work quietly every day, healing the unnoticed, restoring the worn, replacing the lost. Each of us carries within us these reservoirs of possibility. They do not seek attention. They do not signal their presence, but they are there in bone, in skin, in blood, patiently maintaining the body from within. In this way, stem cells are not just biological tools. They are caretakers of continuity, and as you rest, they are at work, replacing blood cells, repairing tissues, rebuilding what has faded, always renewing, always returning to the original script, the quiet, ancient instructions held in every genome. The stem cell does not ask why. It simply knows what to do. It listens. It divides. It becomes a beginning, repeated endlessly in the silence of sleep. Part 16, Cell Aging and Death. As with all things in nature, cells follow a rhythm, a cycle of birth, growth, purpose, and rest. We've seen how cells arise from stem cells, how they differentiate, divide, and perform their quiet tasks. But even the most enduring cell does not last forever. There comes a time when each one must step aside, relinquish its function, and leave space for the new. This is not failure. It is not destruction. It is part of the pattern, the soft, unceasing turnover that keeps the body healthy and in balance. In this part, we explore cell aging and death processes that are gentle, measured, and deeply necessary. Let us begin with cellular aging, or senescence. Senescence is a state in which a cell remains alive, yet no longer divides. It is a kind of permanent pause, where the cell continues some functions, but withdraws from the cycle of renewal. This often occurs after a certain number of divisions, a limit known as the Hayflick limit. Each time a cell divides, the ends of its chromosomes called telomeres become slightly shorter. Telomeres are repetitive sequences of DNA that act like protective caps. Over time, they wear down, and once they reach a critically short length, the cell senses that it should no longer divide. This gentle cessation prevents errors. It helps protect against the risk of uncontrolled growth. It is, in many ways, a safeguard, a quiet recognition that the cell has reached the end of its productive journey. Senescent cells may accumulate in tissues over time, especially during aging. While some continue to serve functions, others can influence nearby cells by releasing signals, a phenomenon known as the senescence-associated secretory phenotype, or SASP. This can be helpful in wound healing, but if not cleared properly, these cells may contribute to inflammation and age-related decline. Thus, balance is key as always in biology. The presence of senescent cells must be countered by the work of immune cells and stem cells, which remove the old and replenish the new. But not all cells age in this way. Some complete their life's work and then undergo a process of programmed death, a quiet, orderly conclusion known as apoptosis. Apoptosis is not random, and it is not violent. It is a carefully choreographed sequence, carried out by a series of internal signals. The cell begins to shrink. Its DNA is gently broken down. The membrane forms small bubbles called blebs. Finally, the cell fragments into neat, contained pieces that are quickly engulfed by nearby immune cells without causing harm or alarm. This form of cell death is vital. During development, apoptosis shapes organs and limbs, removing cells between forming fingers, sculpting the chambers of the heart. In adulthood, it eliminates damaged, infected, or unneeded cells. A cell that detects a fatal error in its DNA may choose apoptosis over propagation, protecting the organism from possible harm. In this way, apoptosis is an act of selflessness, a final service to the whole. There is another form of cell death, less orderly, called necrosis. This typically results from injury, infection, or extreme stress. In necrosis, the cell swells, bursts, and releases its contents, often triggering inflammation. It is not planned, it is reactive, and while sometimes necessary, it is less controlled than apoptosis. Yet even here, the body responds with care. Immune cells arrive to clean the area. Stem cells activate to replace what was lost, and balance is gradually restored. The cycle of cell birth and death is ongoing. In your intestines, entire layers of cells are replaced every few days. In your skin, cells rise to the surface, die, and flake away, while new ones form below. Red blood cells live for about 120 days. Neutrophils, a type of white blood cell, live for only a few hours. Meanwhile, some cells are made to last a lifetime. Neurons in the brain, muscle fibers in the heart. These cells must be maintained rather than replaced, and they rely on careful repair and regulation to endure. The interplay of life and death within the cell is quiet and unending. It is not a loss, but a transition from one form to another. What dies is recycled. What is lost becomes the material for renewal. Even at the cellular level, nature wastes nothing. Scientists are still unraveling the deeper mysteries of aging, how cells change with time, how damage accumulates, how signals become less precise. But even now, we know that aging is not just wear, it is also adaptation. The body adjusts to age as it adjusted to growth-shifting patterns, regulating systems, maintaining the core as long as possible. In each cell, the passage of time is marked not by clocks, but by tiny changes: a shortened telomere, a less efficient mitochondrion, a slower response to a signal. These changes are subtle, but together they shape the rhythms of life. And as cells step down, others rise to take their place. Stem cells divide, tissues renew, the balance continues. So tonight, as you lie in stillness, your body is gently letting go, not of you, but of the cells that have served their purpose. And in their place, new cells are forming, shaped by the same instructions, ready to carry on. There is no fear in this cycle, no drama, only the soft assurance that all things have their time, and that the pattern of life continues quietly, completely, and with deep grace. Part 17. The Microbiome. As we near the final turns in our quiet journey through the living cell, we turn now to something curious, something both within us and beyond us, something made of cells, yet not of our own making. This is the microbiome, the vast, invisible community of microorganisms that live in and on the human body. The microbiome is made up of trillions of bacteria, archaea, viruses, and fungi, most of them harmless, many of them helpful, and some essential. They live on the skin, in the mouth, in the lungs, and most abundantly, in the gastrointestinal tract. There, in the long winding tunnel of the intestine, they form a vibrant ecosystem, an inner forest of life, silent but teeming. These microbial cells outnumber our own human cells by roughly a 1-to-1 ratio, or more, depending on the region of the body. And though they are not human in origin, they live with us from birth to death, growing with us, responding to us, and in turn, influencing the very systems we once thought to be solely ours. Let us begin in the gut. Within the large intestine resides the densest collection of microbes in the body. They are diverse, with hundreds of species coexisting in a balance shaped by diet, environment, genetics, and time. These microbes help break down complex carbohydrates, fibers, and resistant starches that human enzymes cannot digest on their own. As they ferment these materials, they release short-chain fatty acids, which nourish the cells lining the colon, reduce inflammation, and support immune health. In essence, they take what we cannot use and transform it into something essential. Beyond digestion, the gut microbiome plays a role in the immune system. It helps train immune cells to recognize friend from foe, moderating responses and preventing overreactions. Without this early exposure to microbes, the immune system may become confused, leading to allergies or autoimmune conditions. Even the brain listens to signals from the gut. This is the gut-brain axis, a gentle, bidirectional conversation between the digestive tract and the central nervous system. Through nerves, hormones, and microbial metabolites, the microbiome may influence mood, stress, and even behavior. Some researchers have called the gut microbiome a kind of second brain, not because it thinks, but because its influence is so broad, so persistent, and so quietly powerful. Elsewhere on the body, microbes perform other tasks. On the skin, they help maintain the barrier against harmful invaders. In the mouth, they influence the balance of acidity and protect against pathogens. In the vagina, beneficial bacteria such as lactobacillus keep pH levels stable, preventing infection. Every surface of the body exposed to the outside world is touched by this microbial presence. Importantly, most of these microbes do not enter the bloodstream or internal organs. They remain at the surface, forming communities that interact with us without invading. The body recognizes them, not as enemies, but as part of its extended self. From birth, the microbiome takes shape. Babies delivered vaginally receive microbes from the birth canal. Those born by cesarean are colonized by skin and environmental microbes. Feeding also matters. Breast milk contains both nutrients and prebiotic substances that feed beneficial bacteria, along with immune factors that shape microbial composition. As we grow, our microbiome changes. It is shaped by what we eat, where we live, whom we touch. Illness, antibiotics, and stress can disrupt it. Diet, rest, and time can help it recover. And while no two microbiomes are identical, healthy ones share certain features: diversity, stability, and resilience. Like a forest, a healthy microbiome is rich in species, balanced in proportion, and able to bounce back after disturbance. The microbes that dwell within us also carry their own DNA. The microbiome genome, or metagenome, contains vastly more genes than the human genome. In many ways, our biological capabilities are extended by these microscopic guests. And yet, they are not invaders. They are part of us, acquired, maintained, and integrated over time. We do not consciously direct them. We do not always notice them, but they are there, like shadows, like breath, shaping the inner world. Modern research into the microbiome is still unfolding. We are only beginning to understand the full range of their functions, their interactions, and their influence on health. But already, they are changing the way we view the human body, not as an isolated entity, but as a community, a cooperative of cells, human and microbial, living together in silent coordination. The microbiome reminds us that we are never truly alone. Even in your quietest moments, as you sleep, these microbes continue their work, digesting, signaling, protecting, renewing, not as parasites, but as partners, not as passengers, but as participants. Their cells are not ours, but their presence is part of our story. And in that story, the line between self and other begins to blur, replaced not by confusion, but by connection. A harmony of life, at the smallest scale, within and around you. Part 18. The cell in context. As our gentle journey through the living cell draws nearer to its end, we step back, not in distance, but in perspective. We have explored the cell from the inside, its organelles, its functions, its quiet rhythms. Now, we pause to view it not as an isolated unit, but as a participant in something larger. The cell, though complete in itself, rarely lives alone. In multicellular organisms like plants, animals, and humans, individual cells are woven into a greater whole. They are joined into tissues, layered into organs, and integrated into organ systems. This cooperative assembly is the essence of biology's design: many parts, one purpose. Let us begin with tissues. Tissues are groups of similar cells working together to perform a shared function. There are 4 primary types. Epithelial tissue, which covers surfaces and lines internal spaces, forming barriers, absorbing nutrients, and secreting fluids. Connective tissue, which supports and binds, including bone, cartilage, blood, and fat. Muscle tissue, which contracts to produce movement in the heart, limbs, and internal organs. Nervous tissue, which transmits signals forming the brain, spinal cord, and nerves. Within each tissue, the arrangement of cells is precise. Cells are held in place by junctions and supported by a matrix, a network of proteins and other molecules that provide structure and guide communication. No cell floats aimlessly. Each is part of a pattern. From tissues arise organ structures with defined shapes and functions, built from multiple tissue types. The heart, for example, contains muscle tissue to pump, connective tissue for support, epithelial tissue to line chambers, and nervous tissue to regulate rhythm. It is not the presence of cells that makes the heart function, but their cooperation. Organs, in turn, are linked into systems. The circulatory system moves blood. The digestive system breaks down food. The nervous system interprets signals and coordinates action. The respiratory system exchanges gases. The endocrine system releases hormones. Each system is made of organs. Each organ is made of tissues. Each tissue is made of cells. And so, the body becomes a quiet cascade of scale from the smallest parts to the largest patterns. But context is not only about anatomy, it is also about function. A liver cell does not exist in isolation. Its activity is influenced by the levels of sugar in the blood, by hormones from the pancreas, by signals from the nervous system. A skin cell responds not just to its neighbors, but to sunlight, temperature, and damage. An immune cell patrols the body, responding to distant distress calls. In this way, the cell is always listening not to sound, but to chemical messages. It senses its environment and adjusts its behavior accordingly. It is not commanded, it is coordinated. Sometimes, cells must move. During development, they migrate to new locations, guided by chemical trails. During healing, they flow toward a wound. And in the immune system, cells roam constantly, scanning for signs of trouble. These movements are not random, they are purposeful, soft currents in the sea of the body. Context also changes over time. In the embryo, cells divide rapidly, differentiating and forming new tissues. In childhood, cells grow and mature. In adulthood, they maintain and repair. And in aging, some slow down, while others step in to compensate. The context shifts, and cells adapt, always adjusting, always aware. Even death occurs in context. A cell undergoing apoptosis sends signals to neighboring cells, informing them of its departure. Others move in to clear the space. No cell acts entirely on its own. Even in departure, it is part of the whole. And so we see the cell not just as a building block, but as a participant in tissues, in systems, in the body's ever-changing needs. This perspective is especially clear in certain moments. When a wound heals, cells divide, migrate, and specialize. When an infection is fought, immune cells coordinate across the body. When a memory is formed, neurons communicate and change their connections. The beauty of the cell is not only in its parts, but in its placement, its relationships with others, its role in the story. In the forest, no tree stands alone. Beneath the soil, roots intertwine. In the sky, birds move in flocks. In the ocean, coral reefs teem with interdependent life. So too within us. Each cell lives in relationship to its neighbors, to its environment, to the whole. This is the essence of context. And just as the body is made of cells, so too are all living things, trees, whales, fungi, and moss. From the tallest mountain goat to the smallest seedling, life is cellular. The patterns repeat, softly and beautifully, across forms and species. To study the cell, then, is to glimpse the thread that connects all living beings. It is to understand that within every creature, beneath every surface, there are cells working, responding, and sharing in the quiet miracle of life. And within you, they continue now, held in tissues, shaped by systems, responding to needs you may never feel. They work not for themselves, but for something greater. And in doing so, they remind us, even the smallest parts can be part of something vast. Even the unseen can shape the whole. Part 19. Quiet Reflections on Cellular Life. We have walked now through the soft corridors of the cell from its origins in ancient seas, to the intricate folds of its nucleus, to the harmonious work of its organelles. We have seen how it divides, how it ages, how it dies. We've watched it build tissues, form systems, and cooperate in silence. And as we come to rest at the close of this gentle journey, we pause for reflection. What have we truly seen? The cell is the smallest unit of life, and yet In its quiet precision, it contains everything that life requires. Within its boundaries, the entire drama of biology unfolds not as spectacle, but as routine. Molecules move, messages are sent, energy is produced, proteins are built, and all of it happens without noise, without instruction, without pause. Each cell is complete, and yet never alone. It follows a rhythm that is both personal and shared, dividing when needed, resting when full, responding when called. And though its form is microscopic, its presence is everywhere, in the wind-blown leaf, in the beating heart, in the thin film of algae on a river's edge. The world is full of cells, each quietly carrying out the work of life. There is no boasting in the cell, no pride, no resistance to its role. A liver cell becomes a liver cell not because it chooses to, but because that is its place. A neuron sends its signals. A skin cell forms its barrier. And in doing what it is meant to do, each cell contributes to the larger balance. This is perhaps the most humbling truth, that complexity arises not from chaos, but from countless simple acts, repeated faithfully. A ribosome builds a protein, a mitochondrion releases ATP, a membrane lets in water. These actions, by themselves, are small, but together, they give rise to thought, movement, growth, and healing. They give rise to the human experience. And all the while, cells remember their lineage. The DNA they carry has been passed down through unbroken chains of life, from cell to cell, organism to organism, back through time. The instructions you carry were shaped by ancestors you've never known, not only people, but creatures of the sea, the forest, the microscopic world. In this way, the cell is not only a unit of life, but a record of life. And within you, that record continues. Even now, as you lie still, perhaps nearing sleep, your cells are active. Some are dividing, some are repairing, some are simply waiting in soft suspension until they are needed. You do not feel them, you do not guide them, but they serve you, tirelessly, in silence. And they will continue, as you breathe, as you dream, as you wake again, tomorrow. The cell teaches us patience. It teaches us humility. It teaches us that greatness is not always loud, that importance is not always seen. And in this quiet wisdom, there is peace. The human body is made of trillions of cells, and each one holds the whole not in size, but in spirit. Each one carries the same code, the same rhythms, the same ancient pulse. So when you think of life, think not only of the grand, think of the small, the folded membrane, the drifting vesicle, the silent DNA strand coiled in the dark. Life is not made of moments. It is made of molecules. It is made of cells. And it is within these cells that you, and all who have come before, have quietly existed. So now, let the breath slow. Let the mind settle. Let the silence of cellular life cradle you in sleep. And know that while you rest, the living cell, ancient, constant, and full of wonder, continues its gentle work.